the genetic information that codes for the characteristics of every living thing. Plants, animals, bacteria, and you and me. It goes through a process called DNA replication. In the nucleus of the cell, DNA goes through this process in order for growth and reproduction. Let the replication begin.
Topoisomerase, the enzyme that keeps DNA from supercoiling, uncoils and lays out the nucleotides in a way for DNA helicase to follow and easily break the hydrogen bonds between them.
DNA helicase runs over the nucleotides, breaking the hydrogen bonds in between the complementary base pairs so that new strands can be built. The single-stranded binding proteins keep the hydrogen bonds from reforming in between the bases, keeping the replication fork open.
The newly separated strands run anti-parallel to each other, with the leading strand oriented 5' to 3' and the lagging strand oriented 3' to 5'. Enter RNA primase. RNA primase lays down a short piece of RNA primer, represented as the striped nucleotides, as a starting place for the real DNA to be built by DNA polymerase III later.
DNA polymerase III comes along and starts where the primer is, putting complementary nucleotides from 5' to 3', continuously creating the new DNA strand.
RNA primase enters again for the lagging strand. It does the same as before, but instead of just one, it places multiple primers along the DNA. The lagging strand is oriented 3' to 5', and remember, DNA is only built 5' to 3', so it has to work backwards in little chunks called Okazaki fragments.
DNA polymerase III then begins at the primers, adding complementary nucleotides, creating the Okazaki fragments. Then DNA polymerase I enters. It attaches to the RNA primers and replaces them with DNA. It does this for both the lagging and the leading strand.
DNA ligase swings into the process and begins bonding together all of the Okazaki fragments on the lagging strand, completing that strand of DNA.
As you could probably tell from the process, DNA replication is semi-conservative. This means that during replication, the resulting pieces of DNA that are produced are half new DNA and half old DNA. But don't worry, the replication process doesn't end here. It goes on to replicate again, and then again, and then again, and then some more times.
Now time for bloopies! DNA helicase runs over the
I got you for the white one. Go! I got you for the purple one. Hey, ready? Go! Okay, erase! Looks good. Deoxyribose.
this one.
I don't know. What is yellow? What is yellow? Thymine. It's okay.